So today we are discussing about staining technique in microbiology. What is it staining? Staining is a fundamental laboratory techniques used to enhance the make microscopic organisms like what are that cell population or tissue highly visible under the microscopic organisms. Introductions, types of staining methods. What are the methods? And introductions, bacteria or microscopic organisms, so and colorless for the most part of that. Staining is a dye used to color the living or dead organelles, and there are the acidic or basic and neutral dyes used. Acidic are negatively charged and basic is a positively charged based on the dyes also. The and next neutral, and next one, positive staining, simple and differential staining, and structural and special staining, and bacterial stainings also the simple stainings. Positive staining also the simple staining. So simple staining, this technique uses the single basic dye to color all cells uniformly, allowing the viewer to easily observe the size, shape, and arrangement of cells. Two types, positive and negative. Positive direct staining and negative or indirect staining also. So positive is a basic dyes, crystal violet, methylene blue, safranin. Because the bacterial cell wall and cytoplasm are negatively charged, they attract and retain the dye. The cells become colored, the background remain clear. This is the basic shapes of bacteria, cocktail shape and bacilli shape also. And crumb staining is author name is Kongs Christian Josin Crum. While working in Berlin in 1883, he later published this procedure in 1884. At the Dr. Gram was studying the lung tissues. And crumb staining principles. Crumb staining is used to determine the crumb states to classify the bacterial broadly. The crumb staining uses the crystal violet to stain cell walls, iodine as a mordant, and acid fusions or safranin also. This is the dye is a methods consist of the dye. What are that? Primary stain, crystal violet, methyl violet also. Modern dyes, the grams iodine. Next one, decolorized alcohol, acetone alcohol, and counter staining dilute carpal fusion safranin, and also this is the observed for the gonococci also safranin. And this is the crumb staining technique. So the most for gram positive or gram negative, gram positive or purple color, negative or pink color. It also the thickness of the particular layer, the bacterial cell, the four type of the including primary staining is a crystal violet, is a modern staining is iodine, and decolorized alcohol and acetone, and saffron is a counter stainings also. Acid fast staining also known as the Ziehl Neelsen staining. This is a Mycobacterium tuberculosis, or the cell wall waxes are identified the bacteria containing the mycolic acid. Example, Mycobacterium tuberculosis. The procedure requires the heat to allow the primary dye carpal fusions to penetrate the waxy wall. Acid fast cells retain the red colors also. And this is the acid fast staining basic requirements. The primary and modern staining with strong carpal fusions, decolorations with acid alcohol. The acid alcohol containing 3% HCl and 95% ethanol and 20% H2SO4. Counter staining acid are basic cells. The microscopically observation, the stain smear or contain a pink colored slender rod shaped structure are seen in curved ends, and bacilli is seen among the blue colored, and smear is positive for acid fast bacilli. And different modification of acid fast staining. 5% of sulfuric acid is used as a decolorizing agent for staining Mycobacterium leprae. 1% sulfuric acid is used as a decolorizing agent for staining Nogadia species, and Cryptosporium and Leprospora oocytes for modification of acid fast bacilli. This is the third one, structural and special staining. Endospore, capsule, flagella, and bacterial nucleus. This is the special staining. So special staining, specialized techniques to designed identify the specific internal or external structures within a bacterial species. The spores are highly resistant and metabolically so active. Morphological of bacteria endospore is best observed. This is the modified Ziehl Neelsen staining. So endospore staining is a visualize the dormant highly resistant structures For malachite green driven by heat as the primary staining and safranin to counter staining the vegetative cells making spores appear the green and cells appear red or red in colors. So this is the observe the spores. You see the pictures and red in colors, safranin and red in color and vegetative structures are arranged. And principles, the chemically the capsular material is polysaccharide and glycoproteins are a polypeptide. Capsule staining is more difficult than other type of differential staining. The procedures because the capsular materials are water soluble and maybe the is a used. And this is the capsule staining also the for background and the bacterial cell leaving the capsule as a clear. So flagella is the organ of locomotions which is one or more unbranched long filaments. So uses a modern such as tannic acids. This the wet mount flagella staining has been described as a Hambrook et al. In 1989. It is easier, cleaner and more reliable method. And procedure for and this one is also the picture shows the Salmonella typhimurium stained with a modified Rio stain to show their flagella that are peritrically arranged around the cell. And nucleus staining. So highlight the gelatinous outer capsules that protect the bacteria. And this is the cytoplasm has a strong affinity for most stains and is may interfere with the observations also. And nuclear staining in fungi are eukaryotic. Fungal nuclei are often stained with hematoxylin. Giemsa staining is also used in nuclear staining in fungi. Shows the microscopic view of a fungus stain with lactophenol cotton blue. This is standard staining method used in mycology to examine identify the fungal structures. The Aspergillus common genus of mold. You can identify this distinct morphology and conidiospore vesicles and conidia. Thank you.